Title: Use of the supported membrane tube assay system for real-time analysis of membrane fission reactions. Introduction. The introduction begins by highlighting the importance of membrane fission in various cellular processes such as cell division, synaptic transmission, vesicular transport, organelle biogenesis, and sporulation. It explains that membrane fission involves the splitting of a membrane compartment through the application of curvature stress, leading to scission. This process is energetically unfavorable, and cells have evolved membrane fission catalysts, MFCs, to manage it. The introduction then discusses the challenges in studying membrane fission using reconstituted systems, such as the lack of quantitative assays and the requirement for complex apparatus. It also mentions that previous methods, such as perforated cells and purified cell membranes, have limitations and do not provide a clear understanding of the minimum machinery required for membrane fission. The introduction introduces the supported membrane tube (SMRT) assay system as an alternative approach. SMRT templates are formed by extruding a lipid reservoir into long membrane tubes with variable dimensions. These templates are supported on a passivated glass coverslip, allowing for real-time analysis of membrane fission reactions. The SMRT templates overcome the challenges of previous methods by enabling high-throughput analysis of membrane fission, providing spatially resolved dynamic analysis using microscopy-based techniques, and requiring minimal technical expertise. The introduction also explains that SMRT templates have been used to study the membrane curvature sensitivity of dynamin assembly, membrane remodeling reactions, and membrane fission reactions. It highlights the advantages of using the SMRT assay system, such as its versatility, ease of use, and ability to analyze membrane curvature-dependent reactions. Overall, the introduction provides a comprehensive overview of the importance of membrane fission, the challenges in studying it, and the advantages of using the SMRT assay system for real-time analysis of membrane fission reactions. Figure 1 in the scientific paper shows a schematic of the generation of SMRT templates. The process involves spotting a lipid mix on a passivated glass coverslip and hydrating it inside a flow cell. This leads to the formation of large vesicles that are extruded by the flow of buffer, resulting in an array of membrane tubes. The figure also shows the different components involved in the process, including the passivated glass surface, the lipid mix, the flow cell, and the pegylated coverslip. The significance of Figure 1 in the paper is that it provides a visual representation of the process used to generate SMRT templates, which are important for studying membrane curvature and the binding of proteins. The figure highlights the key steps in the process and the materials involved, serving as a guide for researchers interested in using this assay system. Figure 2 of the paper shows the covalent modification of glass surfaces with PEG. The figure includes a schematic showing the pegylation reaction, as well as fluorescence micrographs and quantification of BSA Texas red conjugate bound to piranha-treated and pegylated coverslips. The micrographs demonstrate that pegylation effectively prevents binding of the BSA Texas red conjugate to the coverslip, indicating successful passivation of the surface. The quantification data show a significant decrease in fluorescence intensity on the pegylated coverslip compared to the piranha-treated coverslip, confirming the effectiveness of pegylation in preventing protein binding. The significance of Figure 2 is that it demonstrates the successful covalent modification of glass surfaces with PEG, which is important for creating a stable and passivated surface for the formation of supported membrane tubes, SMRTs. This passivated surface allows for the precise control of membrane curvature and the investigation of how membrane curvature influences the binding and scaffolding of proteins. The data in Figure 2 also provide a baseline for comparison with subsequent experiments in the paper, where the effects of protein binding and membrane fission on the pegylated surfaces are examined. Overall, Figure 2 establishes the foundation for the subsequent experiments and analysis in the paper. Figure 3 in the scientific paper describes the flow cell assembly and characterization process. The figure shows the photograph of the FCS2 chamber connected to a low-adhesion centrifuge tube through PEEK tubing. It also shows a plot of the calculated flow rates as a function of the dial setting. The authors used a dial setting of fine 10 to form SMRT templates and a dial setting of fine 5 to introduce proteins to SMRT templates. Additionally, the figure includes a plot showing the kinetics of increase in BSA fluorescence, which is used to determine the dead time of the chamber. The significance of Figure 3 is that it provides important information about the setup and characterization of the flow cell system used in the experiments. The flow cell assembly ensures controlled flow of buffer and enables the formation of SMRT templates. The characterization of flow rates and dead time is crucial for accurately interpreting the results obtained from subsequent experiments. Figure 4 shows the estimation of tube radius in the SMRT templates. The figure includes a schematic of the SMRT templates with a supported lipid bilayer, SLB, at the source and membrane tubes, as well as fluorescence micrographs of the source at the initial stages of SMRT template formation. The fluorescence intensities of different regions of the source are measured and used to calculate the tube radius. The histogram of fluorescence intensities shows a range of tube radii, with a mean of 16 plus or minus 6 nanometers and a coefficient of variation of 38%, n equals 57. The significance of Figure 4 is that it demonstrates the highly variable dimensions of the membrane tubes formed in the SMRT templates. This variability allows for the systematic characterization of how membrane curvature influences the binding and scaffolding of proteins. The range of tube radii observed in a single preparation highlights the ability to generate membrane surfaces with variable degrees of curvature. This is important for studying membrane remodeling reactions and membrane fission, as it provides a platform for analyzing the effects of membrane curvature on these processes. Other figures that are useful to discuss in relation to Figure 4 are Figure 1, which shows the schematic of the generation of SMRT templates, and Figure 3, which shows the flow cell assembly and characterization. These figures provide important context and background information for understanding the experimental setup and the generation of SMRT templates. Figure 5 in the scientific paper shows the typical results obtained from the use of the supported membrane tube (SMRT) assay system for the analysis of membrane fission reactions. Panel A shows a time-lapse sequence where dynamin, a protein involved in membrane fission, is added to SMRT templates. Red arrowheads mark the regions where dynamin scaffolds constrict the underlying tube. Panel B shows a kymograph of tube fluorescence, where dark and light portions reflect regions under and outside the scaffold on the tube, respectively. Panel C is a histogram of tube radii calculated from tube fluorescence, showing two peaks corresponding to tube radius under and outside the dynamin scaffold. Panel D shows the estimated tube radius under and outside the dynamin scaffold, with a mean of 7, plus or minus 0 nm and 13, plus or minus 1 nm, respectively. These results demonstrate that the SMRT assay system allows for the analysis of membrane fission reactions and the characterization of the membrane tube dimensions under different conditions. The system provides insights into how dynamin assembly and membrane curvature influence membrane fission. This paper describes a method called the Supported Membrane Tube Assay System (SMRT) for real-time analysis of membrane fission reactions. Membrane fission, or the splitting of a membrane compartment, is an essential process in various cellular functions. The SMRT system allows for the creation of membrane tubes supported on a passivated glass cover slip, which can be used to study membrane curvature sensitivity and membrane fission reactions. The method involves the formation of SMRT templates by hydrating a lipid mixture on a passivated glass cover slip, which results in the extrusion of the lipid reservoir into long membrane tubes. The SMRT templates can be used to analyze how membrane curvature influences protein binding and scaffolding, as well as to study membrane remodeling and fission reactions. The method has several advantages, including high throughput analysis, easy visualization using microscopy, and the ability to study multiple fission events simultaneously. Comparisons with other methods demonstrate the superiority of the SMRT system in terms of simplicity and throughput. However, there are some limitations to the method, such as the lack of control over membrane tension and the inability to incorporate membrane proteins. Overall, the SMRT system provides a valuable tool for studying membrane fission reactions in real time. If you are an author or copyright holder for the work presented in this video and you have any issue with the existence or contents of this video, please let me know.